Greetings, I am Gatis Melkus, and I'm here to give you a quick tour of our poster for ISMB ECCB 2023, titled The Utility of Cliques in Topological Characterization of High-C Data. This poster is based on work undertaken by me and my colleagues at the Institute of Mathematics and Computer Science of the University of Latvia. It is a well-established fact that chromatin is organized spatially along both genomic and epigenomic lines in a dynamic fashion, and in recent decades a body of methods has been developed to help unravel elements of gene expression regulation tied to this spatial organization. Of these, high-C and derived methods are highly prominent, and since the inception of the field there has been a corresponding body of network analysis methods used to make sense of the unique topologies observed in chromatin conformation. Today, we present an examination of the functional significance of cliques of size 3 in ensemble high-C data, utilizing 3 publicly available datasets comprising over 40 tissue types. Cliques of size 3, which are triangular graphlets where each node connects to the other two, were counted in all 3 of these datasets, and then analyzed both in terms of their overall count as well as their prevalence in particular parts of the genome. In addition to analyzing cliques of size 3 as part of our work, we also develop a centrality measure to identify links that are part of many cliques, which we call supports, or S as a function of k, where k is the number of cliques that the link is a part of. Since cliques most commonly occur in dense aggregations that are complicated to computationally unravel and analyze, we hope that supports could help us identify links of particular interest, and we'll see how this develops over the course of the poster itself. Intriguingly, we found evidence of consistent chromatin hotspots that were preserved across datasets, most notably in the case of the human leukocyte antigen cluster in 6b21, as pictured in Figure 1. In the case of the HLA cluster, the chromatin hotspot contains the vast majority of the cliques of chromosome 6, as well as a substantial proportion of the links. While not as dramatic as in chromosome 6, a similar pattern of clique clustering in individual peaks was also observed in most of the datasets beyond this. However, these clique peaks, so to speak, were also occurring in areas of high link count, which necessitated testing that this was not simply an artifact of high node degrees in the areas in question. So, in order to test whether these clique peaks are indeed indicative of some specific structure, we conducted a randomization trial in which we swapped link endpoints in our dataset to produce randomized high-C maps with preserved node degrees and link length distributions. As seen in Figure 2, while link distributions were entirely preserved with our method, the clique count, and accordingly the support count, steeply declined to less than 50% of the original count. From this, we concluded that the preponderance of cliques in our data could not be explained purely through increased link counts in those particular areas. For more details about our randomization process, which I won't get into here, you are, of course, invited to consult our other poster at ISMB ECCB 2023, Randomization Techniques for High-C Data. Beyond this, we also analyzed our cliques and supports via gene ontology enrichment analysis, and found that cliques as well as supports were enriched both for hotspot-specific genes, including immune genes in chromosome 6, as could be expected, and olfactory genes in chromosome 14, the site of another hotspot tested in the literature, but also for key cellular functions such as cell adhesion and RNA metabolism, which, while broad in categorization and containing many different genes in their overall category, nevertheless seem to be key cellular functions, albeit ones lacking in any particular specificity. We also employed chromatin annotations to make educated guesses about the potential activity of these cliques, analyzing their enrichment as compared to the links in the dataset for CROM-HMM marks from roadmap epigenomics in the case of our tissue high-C and tissue PC high-C data, and for blueprint epigenomic annotations for our blood cell PC high-C data. As you can see here, of all the marks analyzed, the cliques most appear to be enriched for transcriptional start sites, with an enrichment of 1.5 to 3-fold for cliques and up to 5-fold for supports. There was much lesser enrichment in enhancer marks, noticeable depletion in quiescent chromatin, and primarily either depletion or very mild enrichment of polycomb repressed chromatin. Likewise, in the blueprint annotations, mild enrichment in CDCF was observed, and no enrichment or even depletion of proximal and distal enhancer marks. While it would take more data to prove it one way or the other, our functional analysis is somewhat consistent with what are sometimes described as transcription factories, chromatin hubs where coordinated transcription at many loci is said to occur. In light of this, it is interesting that there is little enrichment in enhancers compared to transcription start sites, and more research is required to precisely elaborate on the functional significance of these hubs, as well as supports in particular, which do appear to refine this enrichment further. Nevertheless, our clique-based analysis does appear to function well in the datasets we chose, and shows remarkably consistent results between them, given the heterogeneity of high-C data. It would therefore be interesting to see if we could broaden our analysis further, and perhaps apply it more generally to a greater array of data in order to identify just how consistent these clique arrangements are, and whether any particular hotspots can guide us to a better understanding to how these cliques form, and what their precise function might be. On that note, thank you for listening, and I hope to see you in the conference.